Okay, thanks. So I give a presentation of my postdoc, Nana Dinh. The title is Climate Impacts of Land Use Change, already presented in ORCHIDEE. And to start with, why are we interested in that? Nana is hired in a project that's called Lamasos. It's also a project where we work with our colleague, Rasha Shakir. And this is a big European project that we want to develop some modeling tools that can help decision makers and people like up to level of EU to decide on policies on land use decisions that are required to reach the goals of the European Green Deal. And that overall project to help that developing of these modeling tools, our task is to create knowledge base with response functions that can predict land use impact on climate, in particular on biosphere carbon stocks. So how we want to do that, we want to use several land surface models, and Lana is working on the model ORCHIDEE. So what we want to do, we will calibrate and evaluate these models first against observations, then run them over Europe to get like a huge set of simulation results and representative for the climate of Europe and for different land use transitions. And then finally, we want to use these simulations to derive response functions that can then be included in these modeling tools that we want to create. But a very important question that we got is, of course, how far are these models, and in particular ORCHIDEE, able to really represent this impact of land use change on these carbon stocks. So this is a representation of how carbon is represented in ORCHIDEE. So we have different carbon pools in the biomass, we have carbon in litter, and we have carbon in like soil carbon. So carbon in the soil is found to the mineral phase. And yeah, of course, these carbon pools are interconnected. So biomass that inputs from photosynthesis, then there's autotrophic respiration, but there's also litter flow that feeds into the litter input. But then you can also have biomass outtake due to harvest, of course, and also when you have land use change. So it's what Daniel just mentioned, that we have, for instance, deforestation in a park. It's part of the forest biomass is taken out as harvested wood. But another part, like roots and leaves, they feed into the litter pool, and that's why what Daniel mentioned, you have after land use change, after deforestation, you have a feed of litter input into your soil carbon stocks, for instance. Okay. And all these dynamics, these changes of these different carbon stocks, they are simulated for different types of vegetation that we call plant functional types. And for Europe, they are more or less four that are important. Deciduous forests, coniferous forests, cropland and grassland. These are the land use types of which we analyze the effect of land use change. And a bit like what Daniel do for soil carbon stocks, we criteria for the changes of carbon stocks in the biomass. It's the biomass of trees, of deciduous and forest forests, and for this we used a database that was compiled by colleagues around from the entire Vienna. And this database that this observations from different kind of trees, and that combines these estimates of the crop grown biomass and the estimates of the age of the tree. And this is what we have here on the right-hand side. We have different classes of tree age. And because for each of these classes of tree age, we observe biomass. We have in black, this is the statistical distribution over the observations from a database. in red, these are the simulations from ORCHIDEE. So what we see is like here, like the average values that fit relatively well. So we can say, okay, that ORCHIDEE is able to reproduce the biomass accumulation in trees over time, at least on average. But what we also see is that for these observations, we have a much larger spread of values, which is of course because these are from individual trees, from very different species of trees, and also very different like environmental factors that play a role, also like related to management that are not represented in ORCHIDEE. There's a much larger variation, but with ORCHIDEE, we have like only the scores, blend functional types like Tibet scores and Ferris scores. So that we are quite happy that we at least these average increases in biomass. So with regard to changes in biomass, carbon stocks, we are quite satisfied. And let's come to soil carbon stocks, because of the line with what Danja are working on. This is like first information how far ORCHIDEE reproduces spatial distribution of soil carbon. So this is compared that for the topsoil against the European database Lucas. And it's all average for each 100 grid cell. So from this we don't see that quite well. But here we have 4 grasslands. We plotted simulations against observations. First, for each half degree grid cell, when you see, okay, we have a fair correlation. It's a kind of big relative error of nearly 40%. But if we aggregate this data further to like 3 degrees, so aggregate by 6 times 6, then we have, of course, much less grid cells, but then we see that the correlation gets better, and also the statistical distributions should be more. And this is because we can only reproduce maybe large scale spatial patterns, which are driven by the climate, but you cannot really reproduce the smaller scale spatial variations that are controlled by other factors. And yeah, so Lana also made a data analysis of observations of land exchange impacts on carbon stocks, soil carbon stocks. So we have cropland, grassland, grassland, cropland, grassland to forest, and cropland to forest. We distinguish observations that only look at the soil carbon from the mineral soils and excluding like litter, the background litter, forest floor, from observations that include that. And here, what we see for the different land exchanges, we also have these observations plotted like time of the land exchange against the absolute change. And black, we have bits for these observations. So this is the bit, the straight line, but the dashed line, we see like the confidence interval. For grassland dropland, yeah, it still works, but here for instance, for grassland to forest, you see that the confidence interval is quite wide, so it doesn't really fit. And what we see is simulations with ORCHIDEE, these colored lines. We get, in most cases, we get the direction of the change, but then as Daniel said, we are totally underestimating the size of the change. Okay, to conclude. So ORCHIDEE reproduces relatively well forest biomass stocks, but it doesn't, of course, represent the large variability what we have in circulation data sets. ORCHIDEE reproduces large-scale spatial patterns in carbon stocks, in far how far they are driven by climate, but not in small-scale variability is driven by other factors. For these land exchange impacts on soil carbon stocks, at least we can reproduce more or less the direction of the soil carbon stock change, but we totally estimate the size of the change, and also as we've seen, for instance, for present force change, the data analysis are actually only of limited visibility to benchmark our model.